Before start this video, please subscribe my channel and click on bell icon. Thanks.
Every year, hundreds of thousands of people in West Africa become infected with Lassa virus, which can cause Lassa fever and lead to severe illness, long-term side effects or death. There are currently no widely approved treatments or vaccines for the disease. Now, scientists at Scripps Research have determined the structure of the critical protein complex that lets Lassa virus infect human cells. The research, published online in Cell Reports, also identified new antibodies that bind to these proteins and neutralize the virus, paving the way toward more effective vaccines and treatments for Lassa virus. This work is a big step forward in our ability to isolate new antibodies to relevant sites of vulnerability on the virus, and it provides a basis to conduct rational vaccine design to broadly protect people against many lineages of the Lassa virus, says senior author Andrew Ward, Ph.D., Professor O. These new reagents described in the paper are already being put to good use and yielding exciting new results. Like many viruses, Lassa virus exists in a variety of lineages, each with slight variations in its genes. This diversity has made it challenging to pinpoint antibodies that recognize all versions of Lassa virus. Scientists have also struggled to isolate Lassa glycoproteins, the spike-like proteins that surround the virus and are the target of most antibodies. In the infectious virus, these glycoproteins exist in complexes of 3, called trimers. For decades, however, scientists were only able to isolate glycoproteins in the lab as single proteins and not in their trimer complexes. In 2022, Ward and colleagues discovered how to use nanoparticles to hold the glycoproteins together into trimers. In the new work, they used that technique to isolate and structurally characterize trimers of the glycoproteins from 4 different Lassa virus lineages. Surprisingly, the glycoprotein structures from the distinct lineages were extremely similar. We were hoping to see more obvious differences that would explain why antibodies didn't recognize all the lineages, says Haley Purrett, a Scripps Research graduate student and first author of the work. Instead, we found a very high level of conservation across the peptide and sugar components of the protein. Using the same stable glycoproteins, Ward, Purrett and their colleagues next used blood samples from patients who had recovered from Lassa virus to isolate antibodies against the glycoprotein. They found new antibodies and characterized previously discovered antibodies that recognize different lineages of the Lassa virus glycoprotein, which may be useful in developing a treatment or preventive vaccine against the virus. The team is already planning future experiments to pinpoint more antibodies against the Lassa virus glycoproteins, as well as further analyzing the protein structures to identify places on the glycoproteins that are ideal for targeting with drugs. Our goals were to not only try and define some of the structural details of these different Lassa viruses, but to provide foundational protocols and resources for the field, says Purrett. We hope our approaches and initial findings help push the science in this field forward. This work was supported by a David C. Fairchild Endowed Fellowship, the Achievement Rewards for College Scientists Foundation, the National Institutes of Health, 1F31 AL 172358, R01 AI 165692, R01 AI 171438, the Thanks for watching. Please subscribe my channel for more videos.